So we have bottom in sight. Let's see, looks to be jumbles. We're going to get set up here before we start investigating. And there goes, I think that was a fish. And because we were late in the water this morning, we are going to move relatively judiciously so that we can make sure that we can get as close to the top of this mound-like structure as possible before the end of our dive. Also, just before we get going, we are going to be taking our second environmental sample, environmental DNA. It's basically a water sample, and from that water sample we will be extracting DNA. It turns out that organisms slough off dead skin cells, or there's just DNA in the water, or their gametes, eggs and sperm. And so we will extract all the DNA from this water sample, and then we will sequence that DNA, and that will then give us a genetic characterization of some of the organisms that are found in this region. And that will just be another tool in our toolbox to help us understand the biodiversity of the Blake Plateau. And I am Alan Collins of NOAA Fisheries and the Smithsonian National Museum of Natural History. We are ready for the next water sample. Yes, thank you. Who's that? Someone call in?
This is John Reed.
Hi, John. How are you?
I'm doing well. I'm glad to see we're getting down there.
Yeah.
Taking any bets if it's a coral mound or not?
I'm thinking it is.
Yeah, John, I don't know if you're seeing the footage right now, but we did just touch down and we're seeing coral rumble.
And a few live, small, probably gorgonians.
Oh, we got a crinoid there swimming?
Yeah, we sure do have a crinoid. It's a crinoid. These are the kind of crinoids that don't have stalks. Instead, it has these little cirri, little sort of feet-like appendages you see at the bottom, and these animals typically climb up and perch on the top of rocks or corals, but they do also have the ability to occasionally swim, as we just were treated to.
Yeah.
I think that's a good idea. we want to make some distance while we can. Can definitely see some small white, probably Gorgonian colonies on the bottom. We get a snap zoom on just in the center, a little bit above center. Yeah. A little bit higher. I think it's a sponge on the... Oh, no, it's a urchin, I believe. Yeah. Yeah, there's quite a few urchins there. This is likely the urchin Okeanos. Thank you. And when you have a chance, one of these white colonies growing on the bottom would be nice to take a snap zoom on so we can... Yep, that's perfect. Yeah. Thank you. I think that'll be enough for people to positively identify this.
I'm not actually sure at the moment if that's a primnode or a bamboo. I can't tell. I'll look back at it later. I mean, I'm expecting that's a primnode, but I thought I saw a node. So.
No, I think we should push on. We're going to make sure again that we can get close to the top or to the top. And I think
Alan, what is your estimated time to leave the bottom?
The time to leave bottom is 3:30. So it's our originally scheduled time.
And about how many horizontal meters to the top?
62 meters to the top.
That's vertical, though.
That should be doable.
Yeah, that's vertical meters. The full trackway is 758 meters,
No.
and that would be to the highest point.
No, it's 315.
Oh.
Sorry, horizontally.
Oh, good.
Yep.
Oh, yeah, I misread that. Sorry. 315 meters to the horizontally. And I was told that I misidentified that urchin earlier. That is a kind of third urchin, according to Chris Ma in the chat. So one of the nice things about this, the way this whole NOAA ocean exploration is set up, we have a lot of shoreside scientists who are following along at the same time and helping to identify what we see and providing information to the biology leads, such as myself. It's another one of those urchins. Is it convenient to get us, or it looks like we're moving past? Yeah, yeah. Yeah, let's take a snap. That'd be great. And maybe Chris Ma. Oh yeah, I can see that looks a little different than I was expecting. You can see its spines and its 2 feet, nice with that beautiful close-up. Hello.
Hey, this is Chris Ma.
Hey, Chris. How are you?
Hey, Alan. Good to hear your voice. And nice to have you back after such a long pause.
Yeah.
So this, based on what we looked at, what we've seen before on dives to this area, this looks like the kind of therid called hygrosoma. And one of the things that is very distinctive about echinotherids that I love to point out is the fact that, if you remember from the close-up, there are little spines that have little horseshoe tips on the ends, and they use those to assist them walking around on the bottom. Echinotherids are often referred to as pancake urchins or tam o' shanter urchins by the New Zealanders because the water actually deflates. When collected, the water escapes from the coelom, and they flatten out to look very pancake-like. They are actually rather venomous. I forget that's not the right word. The spines can contain very painful poison. So if these were to be collected, for example, and I'm not saying that we would, but from past experience, people who have collected these have had been jabbed by the spines, and I'm told that the sensation ranges from very painful to just sort of a numbing feeling, and it can go through gloves. So and these are related to the shallow water fire urchins that you find in the Indo-Pacific. So we might see 2 or 3 different genera of fire urchins in this area. So I'm looking forward to seeing them. They're always very attractive-looking animals. So awesome. But then, of course, jellies and that swimming shrimp are no slouches either. Anyway, thank you. Looking forward to.
No, I was just going to say we did get a great look at a beautiful surgested shrimp swimming by as you were finishing up there.
Surgested. Lovely. Yeah. Great. Thank you. And,
thank you, Chris.
Looking forward to the dive.
Yep.
Bye-bye.
So, Chris, this is John Reed. I can attest to the potency of that venom. I managed to get jabbed by one a long time ago. How are these different than Areosoma? A long time ago, we've collected Areosoma on some of the reefs. They look similar. They're both the Hydrosoma type, where the
wow, that's a nice view of this fish. I think Chris Ma dropped off, so he can't answer your question, John. But we are getting a great view of a small fish from the water column. Colloides, says Kevin Rademacher from the. It's a viper fish. We could take a look at that yellow fan just a little bit to the, it's 3 o'clock. You can see it. Get a nice close-up on that. As you can see, as we're moving up the slope here, we're finding more and more larger organisms. Ooh, and there's glass sponge just to the left of that, which we'll also like to take a look at. This is a gorgonian, but I don't want to say with the wrong group.
I just stepped out for a moment. Sorry about that. This is an Acanthogorgid, I'm pretty sure.
Acanthogorgid. Yes, please. Glass sponge would be great.
It's relatively tall, skinny polyps, and it's got that dark axis.
Mm-hmm.
And then this white gorgonia in the front, and I'll let you talk about the sponge.
Yeah, this is a glass sponge in the group Euplectellid. It has a sieve plate, a very, very thin sieve plate. It's reminiscent of the genus Euplectella, but it's not that. It's going to be an allied genus. Now we're looking in at a brittle star with a pink disc. And we will want another good close look at one of the smaller white fans when we have a chance. So we're making some good progress. I get the sense that we're ascending, but we're still at about 810 meters. You can. This sort of looks like a barren landscape, you know, from afar. But I'm quite sure if we zoom in on anything, we're going to see so much. Perhaps another viperfish in the water there. So I think it would be fine if you were to pick a spot anywhere in the rubble to maybe get a close-up, and then we'll see if we can look at some of the smaller organisms that are inhabiting the interstices, the little spaces between the rubble. Yeah, I think that's fine. I imagine that pretty much everywhere we look, we're going to find small sponges encrusting the dead rubble, and yep, that's great. Oh, and there's a little white fan we can take a look at that's just below where you're looking. Yeah. All right. Yep. We're a little on top of it.
Yeah, this fan must be a Primnoidae.
A Primnoid?
I think so.
And you can see in the background lots of the rubble there. I think that's good for the Primnoid.
The rubble is definitely Lophelia rubble.
Definitely Lophelia?
Yeah, I'm pretty sure it is.
Great.
There's some solitary coral in there, but by and large, it looks like Lophelia.
Just anywhere in the rubble. We can see a little white sponge there. Yeah. Yeah, take a quick little zoom and just see. Now we can see there's a bunch of hydroids that come into view. I think those are hydroids, or maybe Oh no, actually those are probably little crinoids underneath there, and you can see the white sponge. And you can see green sponge and yellow sponge. It's just to show that the rubble, although it kind of looks dead, is actually full of a really high diversity fauna. A fauna that we call the cryptic fauna sometimes.
And Alan, as we were zooming out, there were two corals there flanking what we were looking at. The primnoid was the fan on the right, and it's some kind of plexor that's much more open branching on the left. So there's at least, I think I've seen at least 4 different octocoral species so far.
Oh, great.
Already.
Oh, and this is, if you have a minute, if it behaves, you could take a video of this. We collected this once early on in this trip, but we didn't get great video of it. So this is a little benthopelagic medusa. No, it's, I'm trying to decide. Wow. It is all tuckered out. I'm wondering if it's Tychogastria. Or something related to it. So I do think this is what I collected before, but I think I may have misidentified it. Yeah, it very much looks like the genus Tychogastria, which is a well-known medusa group that spends a lot of its time on the bottom.
Well, and my go-to is always benthocodon. I think it's the same family or a different family.
It's actually a different family, but benthocodon also spends a lot of time on the bottom, but they are closely related families. Yep, all good, great. So you can see these white vase-like sponges are actually quite common, these euplectellids. Yeah, that looks like a good spot for snap zoom as well. If you, yeah. All right. Little. Okay.
A lot of secondary colonization, huh?
Very much so. A little taller sponge to the left there, a nice demo sponge, and another little white fan. You can get a close-up on that fan. That'd be great. Help Scott identify that. Yeah, if that's...
I was actually focusing on that pink thing behind
Oh.
it, and maybe well, this is good. This is good. I was just wondering if the other thing was a compressed soft coral nephthiid. Yeah, what am I looking at here?
Do you see nodes? Matt?
I don't see nodes.
I don't see any.
I don't think it's a primnoid, so it's got to be a plexorid, I guess. I don't see any scales.
Mm-hmm.
It's got a little...
The shape of the colony and the branches suggest primnoidae, but the polyps don't suggest primnoidae. So we'll study that later.
Biology.
The thicker white one, interesting. Yeah.
Yeah. And then I see the pink organism that looks like a soft coral. Yeah.
Yeah, it looks like it's contracted, right?
A nephthiid with its polyps pulled in.
Oh wow.
Yeah, we don't usually get to see them compress. This is great because I was corresponding yesterday with the web gurus and telling them why I didn't like the name soft coral for all octocorals. This is a soft coral. It doesn't have an internal skeleton. It fills its branches with water. But here you can see it's compressed and released that water, and so it's all reduced. Good example of what a soft coral can do.
Mm.
It looks like that was a nephthiidae. Would you agree?
Yeah, absolutely. Would agree with that, John. In that family.
These little white finger sponges, they kind of remind me of Petrosiades or Petrosides, but I'm not sure.
Okay.
I think Chris left us.
Is that a Chrysogorgia?
I think this is a Chrysogorgia, yeah.
Ah, very nice. With Uropticus in it, one of the squat lobsters.
Yeah, there is a Uropticus, yes.
Hiding right in the middle.
It's funny, in my head I was saying that word Eurotychus, but Uropticus makes sense.
So that's how I always read it too.
You got to watch out with me because next time I probably will say Uropticus and, all kinds of different ways. Eurotychus. I don't know. It just depends how you break up the syllables.
Right. Well, there we have a little, I guess that's a rat tail. Off in the distance. Maybe not. I'm not very good with fish, obviously. But again, one of the nice things is having this high definition. No, that's, boy, it looks really engorged, very full.
Yeah, whatever this is, it just ate, I think. I don't think it's a rat tail.
No,
it is. I'm not used to seeing a tail from that direction.
Yeah. You can look at that little red colony there. I think that might be Anthomastus-like soft coral. If soft coral is the right word to use for Anthomastus-type corals.
Yeah, that's good. They're related to them, but these are definitely getting stiffer, but they're not inflated. So if my definition of a soft coral is that it has to have no axial skeleton and be able to deflate, then this would not be one.
Okay. Oh, that's a nice looking one. Looks like a cupcake with all of its really, really tall polyps sticking out of it.
Yeah. Yeah, these Anthomastus and relatives are really interesting because of the size of those polyps. They're really quite large, like the size of our fingers, But that sort of toadstool-like base that it's coming out of, it's kind of like a mushroom, is really quite cemented. It's got lots of sclerites and tissue that are all kind of put together like concrete, and then there's all the tubes that are running through that are interconnecting the guts of those different polyps. Nice. So that's a lot stiffer than the soft coral that we were just looking at before.
Could you zoom in on the sponge that's just behind this?
More closely related to the bubblegum corals.
Oh, really?
They are too.
And then just a little bit lower, it's a spiky white ball. Sorry, Scott, I'm also...
No problem.
Yeah. That looks very much like the cladorizid sponge that we collected recently down in the Straits of Florida, Abyssocladia.
And just behind that, a bamboo coral, and behind that, maybe a corallimorph.
Oh, yes, and then also a corallimorph polyp, also just a little bit up and to the left from where we're seeing. You can take a little zoom right in there. Yeah, and this is essentially, sometimes they're referred to as naked corals, but this is the sister group to the scleractinian corals, and their main anatomical difference is that they do not have a hard mineralized skeleton. Yes, please. We're gonna
forward we'll go.
Yep, we want to keep making good progress and see how much, if we can get to the top of this in our limited time.
So did we truly make 820 meters when we got to the bottom?
I think we started just shy of 820, yep.
Okay. So about 20 meters up the 50-meter mound.
Mm-hmm.
So we're seeing already so much diversity and not a big density, but it's not bad. But can you imagine where the strong currents are, what it's going to be?
Mm-hmm. Yeah.
Yeah. I mean, based on what John suggested, we're in the shadow of the current. So this is not the optimal feeding place. This is the suboptimal feeding place.
Organisms are doing pretty well in the suboptimal.
And seeing these Lophilia mounds, it makes me think of like sand dunes, and I wonder if like the grasses and sand dunes that help sand accumulate, if Lophilia plays a similar role like that. Just keep on building up substrate that way with their skeletons, plus the sand accumulating.
Mm-hmm. take a zoom in anytime again when we're... No, let's keep going. Thanks. Sorry about that. Apologies. Oh. No worries. I think we'll just keep going, yeah. So have to wow. And looking just now at that angle over that, when you can see over the edge of the hillside there, how many of these different large fans. Let's take a look at the largest one over there. That would be great.
I believe that is a bamboo coral.
Yeah, that is definitely a bamboo coral. We can see the nodes. Now the splitting does not happen at the nodes. The splitting of the branches.
I'm fooling you guys. This here. Yeah, this is kind of a classic genus Keratoisis morphology on first glance here, with those branches arising from the internodes. That is those white bits between the dark protein nodes. And you can see it's a nice planar colony. All the branching is in one plane. Pretty thick, robust polyps. Very nice.
Scott, why do they have those black nodes when something like a Lophilia does not?
So this is the ever-present question of why when we're talking about evolution and evolutionary history, and it's a little hard to predict why something did it, but we can come up with some suggestions about how it might be functioning now. But this is in the, if we look at the Cnidarians, you've heard Alan talking a lot about the hydrozoans. That's a class. There's another class, the scyphozoans. Those are jellyfish. The class Anthozoa is where you have all of your more traditional corals. And they're divided into two groups, hexacorals and octacorals. The hexacorals are the Lophilia you just mentioned, which is what's creating the huge mound and much of the rubble that we see. The octacorals are a little bit different in that they have many of them. Maybe we should talk about the fish first, but I can come back to that.
Not sure what to say except that it looks very pretty. Very sleek eel.
Synapobranchid eel, I think.
Synapobranchid?
I think so.
It is a cutthroat eel, yes.
Yeah, just quickly getting back to the octacorals, they have an internal skeleton, so their skeleton is kind of like our own, and it's completely encased within our body. Not so for the scleractinians, where that skeleton is actually external to the tissue. And in at least the bamboo corals, for some reason, the protein-based skeleton has been replaced a lot or overlaid with calcium carbonate. And so that's that white that you're seeing. And it's broken up so that the protein underneath it still shows at those nodes. And the suggestion is perhaps that makes it a little bit more flexible instead of just having a purely calcareous skeleton. I think the jury is still out on that pending the data, because inside of that protein node there is a little bit of the calcareous stem connecting the two internodes. So I think the answer to your question, Matt, is I don't know.
Let's look at that. Let's see if we can get...
Wow, it's almost invisible.
It is almost invisible. You can see how jellies... I think it was a Narcomedusa of the Solmissus group. Yeah, it's definitely Solmissus. It's just really hard to get the contrast on it here. That's beautiful. Okay, but we got to remember this is not a water column dive. So. Oh, but
it's amazing.
That really is. That was beautiful. It had a lot of more tentacles than most of them. And I think there goes, yeah, snap. While we're here. Oh, nice.
So maybe
That's perfect.
Maybe the currents are picking up here.
Yeah.
We're seeing the planktonic organisms floating by. Oh, that first one though, it looked like they took the transparency slider and just turned it way up.
Exactly. Time to disappear now.
Alan, any idea of what that one was?
The second one was more of a, I think is a Minthia. I think we're in Rhopalonematid country. That's a family Rhopalonematidae in the order Trachymedusae. Trachymedusae. Oh. Thanks. Great. Let's take another zoom in on some of these smaller white colonies. I don't think they're all the same. And we had some requests from the chat room to take a look at some of the smaller. Oh, what do we have there inside that?
It's like an anemone.
Yeah, a little flytrap anemone.
Yeah.
Get a nice little look at it.
Always photogenic.
Oh, yes, very photogenic and fairly commonly seen on these. Oh, look at the texture of it.
Yeah.
Wow. I like that.
Huh. Nolan Barrett says the dissolved oxygen is relatively low here. Yeah, the oxygen minimum zone in the Blake is, I believe, not very common because it's so shallow. You don't see that as much.
Mm-hmm. Thanks for that zoom.
Really loving this body wall.
Yeah, I don't know what to make of it. It's not something that I feel like I've seen much of. And so we can just pick out some of these smaller colonies and maybe snap from one to the next. Yep, that works. Okay.
This is a Plexauridae, family Plexauridae, the white one, and I'm pretty sure we collected one or two of these back in 2019. So hopefully someone gets an ID on that soon. And this is another of the soft corals, but it's extended now.
Yep, Nephtheid. I think that's...
Oh, and I think I'm seeing a skeleton shrimp, a caprellid, just in the background there. Now it's going off to the right on the Lophelia in the background.
On Lophelia? See if we can zoom in.
One of that brownish, broken, dead skeleton back there in the background. It's on the to the right of that brittle star that's in.
Just to the right of the brittle star? I think you're going to have to zoom. Usually they're pretty small.
On the tip of the brown, dead Lophelia
oh, yeah.
to the right of the brittle
there
star.
It is, standing on the... It's long and thin, right on the tip of the Lophelia. And you can see it bending from... It's bending back to the right and then down. You're straight at it. It's
Wow.
very, very thin. That's good eyes, Scott, that you would see this. So this is a kind of amphipod that they wind up perching. They're called skeleton shrimp, and they'll bounce back and forth, catching organisms. It's super tiny.
It's like a little praying mantis on the
yes,
front end of it.
Exactly. It holds its little keely up.
Little nodders.
Yep. Nice job, Scott.
I mean, it's that whole it's what draws your eye to them. That's how you can find them.
Yes, we're good to go.
All over the place.
Let's see another look at that.
This is great. This is the sort of thing that makes the annotators, the video annotators, pull their hair out. So we have a team of students who are trying to make observations, keep up with observations, or doing it post-dive, and there's an awful lot of stuff in there to make a notation to.
Right.
Which we love.
Sure, we'll take another. I think we've already seen it. Yeah. It's a nice little... I can see a different anemone there that's in pink just to the... Yep, exactly, 8 o'clock kind of thing. Yeah. And then when we zoom in on it, we're going to see all sorts of things right next to it. That is nice. I think that's good. Oh, a little closer. Nice.
The morph we see out here quite a bit, and I'm still not certain of what the name is.
Mm-hmm.
Olo something.
Hey, Alan, how's it going? Sorry, I was late.
That's all right. You had important things to do.
Yeah, I was helping.
Little outreach.
Yeah, I was helping Miss Flynn's high school class
Nice.
and talking to her students.
Yep.
Anything fun? I saw the corallimorph.
Yeah, there have been a lot of
jellies. They were cool jelly stumps.
Really
I was
cool
watching
jellies.
I was watching downstairs while I was talking. So yeah, this looks like it's going to end up being a Lophelia reef.
I think it might be a Lophelia reef. We've seen
Yeah.
lots of dead Lophelia and lots of things growing in the rubble.
Awesome.
In and on and between.
Yeah, the huge habitat for many species of animals.
Yes.
Lots of gorgonians. It's like a field of them.
It's
Pretty cool.
very cool. And we've been seeing some nice glass sponges. I think it's maybe Heterotella or something. It's a euplectellid with a very thin sieve plate on top.
Oh, nice.
Very delicate. Although we should probably take a look at another one because we've only looked at one of them closely. And then I've sort of been assuming they're all the same, which is not necessarily the best assumption. I think, yeah.
It looks like it's getting a little steeper, maybe 30, 40 degrees when you look sideways. So
Yeah.
I expect to see the live coral very, very close to the top. It's not going to come too far down on this north slope. It'll be really, really good to get on top and partway down the south slope. If there is live coral, that's where it's going to be.
Okay.
Yep. So the core rubble on the sides of these mounds is totally normal. It's not like this is some kind of, ecological disaster or something. It's
Quite
the way
the
it
opposite,
grows.
right?
Yeah, so it's totally normal, healthy. The way these bioharms form. Did we go over this yet?
A little bit, but
Okay.
a little rehash.
Meh.
Corals grow, they die, sediment falls
Fills in,
in.
and then they grow more.
Yes,
Yep.
they grow on top, and they create. Is that one of these tubes right there?
Yeah. Can we zoom in center screen?
Yeah, I just want to check out this this euplectellid again. Yep, that looks pretty much similar to what we saw last time. It's nice. It's interesting how common they are. So this is the kind of glass sponge where sometimes little shrimp are associated with them.
You can see them right there.
Yeah, right, you can see it?
Yep, there's one right there. There's a little shrimpy.
Yes, and so they will get inside the sponge as larvae, a male and a female. Oh, that's
They're
really
both
nice.
in there.
Yes, and they will live out their entire lives protected inside this sponge. And they've got, it's like a little cage. But I always find it interesting that this is sometimes a traditional wedding present in some cultures, as I guess symbolic. But these sponges get in there when they're really tiny. I mean, these little shrimp get in there when they're really tiny, and then they'll raise a new brood right inside there. That's great. Yes, thank you. That was great to see.
Looks like some standing dead now starting to appear. A little bit of standing dead coral. It's right at the bottom here.
Mm-hmm.
Yes, we're coming up slope. How far are we up?
We're up to about 780. We were about just shy of 820. So
40 meters roughly.
yeah, we've almost come up 40 meters, which is great. So because I know we really wanted to see the top of this, and we have limited bottom time today.
So we have another 30 meters or so till we get to the top of this mound.
Yeah, and I believe about an hour of bottom time.
So I see the top. Looks like the top up here. So that's Chaseon, it looks like, at the upper right. Chaseon fenneri, which is the golden crab. There's a fishery for these out here.
Nice.
That's cousins to the one we saw the other day, which was the red crab.
Yep.
Chaseon quinkadins.
And then we have another urchin there. That's pretty much dead center. We're doing a pilot change. I don't think that's a Echinus. You know, we saw something early on that was called Hydrosoma.
Oh, okay.
It was venomous. Spines are venomous. And this looks a little bit like that. It's a little more flattened.
Pancake. Yeah, pancake
little
urchin.
pancaked. It is, when it's collected, it goes really flat.
Yep.
So we know very little about the age of these Ophelia mounds in the Western Gulf, Western Atlantic. Some of the coral that's been collected, some of the larger dead Ophelia that's exposed on the surface has been aged up to 35,000 years old.
35,000?
Of course, nobody is cored.
Hey, Alan.
Hey.
No one has cored these mounds on the west side, on the Western Atlantic, but they have cored a mound off of Norway, a 100-meter mound, and they cored from the top to the bottom, and the base was 1 million years old.
Yeah. It's a really slow-growing species. It grows between 1/3 and 1 centimeter a year. So you can imagine how old these corals are, how long they've been growing there, and then they've been here dead for quite some time in order to collect this ferromanganese crust that forms on them and turns them this dark brown or black color.
So, Alan?
Yeah,
I
Chris.
just wanted to amend something that I said earlier.
Please do.
Yes. So you're correct. I mean, they're definitely Chinothurids. But I did check my notes comparing what John Reed had said about having seen Ariosoma in this area, and I was also looking at the distinction between them. It is very difficult to tell based on anything but the specimen, but in the past, when we've been in this area, we have seen Ariosoma, and they do have that pinkish kind of color highlight to them. I think this might actually be Ariosoma rather than Hygrosoma, which is more white. To be honest, a lot of the distinctions between these urchins is extremely difficult unless you have a specimen. And since I don't want you to come home with, a bunch of urchin needles stabbed in your hand, I think we can leave them as Ariosoma for now. And one of these days, maybe I'll figure this out, and we don't have to be quite so. We don't have to be quite so ambiguous about it, but anyway, thank you for allowing me to indulge in my various little taxonomic musings, and I'll let you get back to it.
Thanks, Chris.
Bye-bye.
That's a neat one, that glass sponge.
Yeah, the glass sponges.
I think it's two next to each other.
Yeah, and they're just sort of bent in the same direction. Oh, that's another nice-looking one. And I think what I was thinking vis-a-vis collecting would be nice to maybe hold off completely until we get to the top.
That sounds good to me. A lot of these
And
species
then-
will repeat. You know, there's
Yeah.
usually more than one.
And even if they don't, so one of those jellies we saw looked very similar to the one we collected, but I think maybe the one that we got was a juvenile.
Oh, cool.
And I have it in the wrong. I have my ID quite messed up.
Uh-oh.
But we've got the specimen to look at.
Here's a pencil urchin. Have you seen one of those yet?
No, we haven't.
Okay,
A
so-
Sideroid?
Yep. That's probably Eusideris. Can you zoom in on the pencil? There you go, right there. Thank you. So I see the top of the mound, and I am not seeing any living coral on this.
Mm-hmm.
Now there's other. Is this in a multi-high mound? I thought we were on.
Yes, this has, I think it's like 3 or 4 different
Okay.
peaks.
There we go.
There's a nice...
I would call this Eusideris. Of course, Chris Ma is more than welcome to chime in and yell at me.
He says it's not Eusideris.
Oh,
Maybe
owned.
He said that before you even said it was Eusideris.
Oh, man. Darn it.
But
Yeah, the
Stylosideris.
probably.
Stylosideris.
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 Yeah, the one we used to see, Stephanie, has the shorter, stubby.
Yeah, that's Eusideris.
ooh, that colony right to the left of this Eusideris is something that looks very similar to what Steve Cairns has requested. Someone who's probably published more new species based on Okeanos material than anyone on the planet. Yeah, except I would like to get a little feedback from Scott France if he thinks, is that a small corallid, Scott? Steve said we wouldn't notice it unless, we were zoomed in and it was just going to look like a, pretty much something like that. I think maybe we've lost Scott.
No, I had to write
Oh.
down. I was in the wrong lab.
Yeah. Do you think?
I don't think that's the. He's looking for Distichoptilum, right?
Yes.
I don't think. I think that's the small Gorgonian
Okay.
sword that doesn't have more full of.
Thank you.
Good eye, though. I'm glad you're thinking about it. I forgot about it.
Yeah.
We're good, guys, whenever you
We
want.
can keep going. Thank you.
There's lots of rippling in the water, huh? Temperature
Yeah.
changing?
Once we get to the top, I'm not sure if it's a temperature
or
or.
salinity?
Yeah. Must be
Pilots were
temperature.
noting the temperature was increasing as we came up the slope.
Yeah, we're close to like 9.9 degrees right now.
Uh-huh.
It's
And we're
within.
up to about 777 meters. Yeah, the temp, 10 degrees, that's pretty warm.
They can tolerate up to 12 degrees Celsius, from my memory. The Lophelia
The
can.
Lophelia, uh-huh.
So, and I think down to 3 degrees is that temperature range there.
So I think our dive track takes us to the top, and then sort of over a little lip, and then to the highest high on the overall mound. It's that little flop. You see an octopus?
I see a star. No,
False, it was an enemy.
anemone. It's like a corallimorph or something.
So lots and lots of these small plexaurids.
Yep.
Those anthomastus-like colonies are really standing out with the bright red.
There's some yellow here, too. Those yellow gorgonians.
Yellow.
We're here at 6. Yeah, go for it.
Sure. Is this a Canthagorgia?
And look at that huge solitary cup coral next to it.
Oh, yeah.
It could be a Canthagorgia.
Unfortunately, when I ran down from the other lab, I left my glasses there. But yeah, this looks like the Acanthogorgia again.
Yeah, Acanthogorgia. We're good,
That's
guys.
that
Thanks.
strange white one to the left.
The one to the left that's white?
Very small.
On the left, I think
polyps.
a primnoid in the
Primnoid?
foreground that is feather-like, and then a tipped-over plexaurid, I think, in the background there. Not 100% sure. Remember, Stephanie, I said the other day when we get up shallower than 800 meters, I'm on more tenuous ground.
Yeah, right.
That's what I meant.
Well, right at the limit of your knowledge, huh? 775.
There you go.
Maybe a bamboo car out 3. Yep.
Oh, yeah.
The primnoid next to it.
I think
It's like plexaurid.
plexaurid is she's interested in.
Okay.
Oh, and you see a little Pharea or Euretid
Sponge?
glass sponge to the left. The little fluted guy.
Little squat
Looks
lobster at 6.
a little chunky. That's definitely not Pharea. But
This one in front of us?
yeah, but it's similar.
It's got something living in it.
Who's in there?
Oh, yeah. Crustacean or something moving around. Maybe a worm.
Right at his base.
So while you're looking at the bunch, the two white octocorals on the right, you see one has all the polyps withdrawn, one is branched and has polyps out. What you saw before, Alan, you thought maybe that's what Steve was looking for. I think was just that.
I got
so
you.
If I'm wrong, then all bets are off. But I'm gonna try to look at that species that he's interested in.
Okay, great. I trust you.
Here we go. What's that? Come on out, bud.
Peek out.
I think it's a crab or shrimp or something. And we just saw his chelate hand, chelate arm.
Thanks for trying for it.
Yeah. That's awesome.
Good approach, pilot. Great.
Yes, we're in turbo mode. So I have a hybrid car, and it's got a turbo button.
Yeah?
Yeah.
Does it have an eco button, too?
Yeah, it has a eco button and a regular and a turbo. I leave it on eco.
Yes.
Unless I'm on the highway, then I'm like, yeah, turbo.
Yeah.
But it's a turbo button on a hybrid. So.
I'll take a snap on that white thing. What is that?
Yeah, it's another.
That's another.
Ariosoma?
Yeah. Ariosoma. Thank you.
Pancake urchin, we're good.
Oh, and those.
Yeah, you can see the poison sacs on top of some of those stems there, on his little spines. It's getting steeper. We're coming up on another high.
Yay! Always.
Yeah, we're looking south now, so.
I like your view as well. As you come up the saddle here.
Likely if this is a living reef, in parts, most of the living coral is going to be on the south side of the mound. Ooh, what is that? Oh, a jelly.
Yep, a ctenophore.
Little shrimp going by in the main view. Yeah, now that one's squishy.
That one is pancaked.
Deflated.
Yeah, they have the ability to suck water in and inflate themselves. And
there's a white sponge. Have you looked at one of those yet?
Here, you mean?
Yeah.
No, we haven't. Maybe once we were looking at. No, I don't believe we've looked at that. So the large white, yeah, exactly. Get a good look at that.
Yeah, as long as we keep moving as quick as we can snap zoom in between.
Nice little fish there.
Nedzumia, a rat tail. You can see when we zoom in, there's 5-8 species, different things, a couple sponges, some
yeah, a lot of
Fangorgonians.
sponges. Oh, I like the little white sponge there that's just to the right of this fish.
Yeah, he's really round with a white membrane on top of the oscule.
I wonder if this is Leucopsacus.
I have no idea, Alan.
Neither.
I don't think I've even heard that
I
species
have little
before,
names.
sir.
Well, there are estimated to be about 1 million marine species, and we have named and described about 250,000 only. So
and my brain retains about 30. So
yeah, so we still have a lot of work to do in terms of characterizing the life in the ocean.
So that's a big yellow sponge. Maybe Pacastrellid. Let's see. What do you think? Dendroceratid. Very lumpy.
It's a demo sponge for sure.
Demo sponge.
Demo, yes.
Ooh, a yellow one.
What was that yellow colony?
That's cool.
That,
Yellow Gorgonian. It's very pretty.
Oh, I see. We're seeing it just on its planar surface. I thought it was
a single stalk,
like a bottle brush.
Yeah.
Yeah,
it's probably the same species. It's just that we can see it from the side now.
See
look at that crinoid.
Yeah, that's a larger one.
Is it a stalked crinoid? I think that's a stalked crinoid. That looks a lot different than, See how far away his roots are. Go ahead, John.
I try to get a good shot of that because I think Chuck Messing is on this.
I was watching.
Viewing this right now.
Okay. Hi, Chuck.
I hope so.
Are those at Siri, though, on the right-hand side?
Yeah, that's why I'm saying I don't think it's a cumatulid, right? I think it's a stalked crinoid because down here at the base
that's a long way away.
Yeah, it's pretty far away from the mouth. So it's probably a stalked crinoid.
And there are at least some of these stalk crinoids that do have those Siri-like extensions going up along the stalk.
Mm-hmm.
Yeah, I was working with Chuck Messing in the Bahamas a few months ago. We were in a Triton sub northeast of Little Bahama Bank. And, of course, his expertise is stalk crinoids. We were seeing endoxocrines and neocrinus.
Oh, nice.
And some other crine.
Yeah, I can't keep them all straight. This one's definitely a stalked crinoid. So they are limited to the very deep sea. They're now no longer in the shallow waters, but they were at one time, like during the Burgess Shale era, eons ago.
Little post-Burgess Shale, but yes.
Post-Burgess Shale, okay.
Paleozoic.
Yeah.
Yeah.
I'm close.
That's it, yeah. Absolutely.
Some of my data's right.
They have crinoid fossils all over Kansas or something like that.
Yeah.
So there was nice anemone there, too.
Yeah, anywhere that was shallow water during that time period has a lot of these really neat stalk crinoids, shallow water stalk crinoids.
This is State Gem, I think, or one of those states.
Is it really? That's awesome. That looks like a bubblegum coral dead. That's odd.
Yeah, that's definitely what that is.
Where'd that come from?
I mean, it could have been growing just in situ here, and because this rubble is relatively loose, that it's been tumbled over or knocked over maybe by passing fish or something.
Hey, this is Chris Ma.
Hey, Chris.
Hey, I just wanted to comment on something that that was interesting based on what you guys have been seeing. So those stalked crinoids that you've been finding, and you're right, there's something like endoxocrinus. The thing about those is that a couple of years ago, Tom Baumiller and Rich Mui and a few others found that sideroid urchins feed on those. And we actually saw one in progress feeding on one a few years ago. And they attack them by going after the tail. So these stalked crinoids can actually move, unlike other types of stalked crinoids, which are anchored to the bottom with a permanent holdfast. These particular ones end in sort of a tail with a bunch of cirri coming off of them. And so the crinoids come after them and sort of munch them from tail first and go all the way up to the top. We just saw some sideroids that might be a possible predator, so it's possible that those stalked crinoids might have been the recent evidence of predation by sideroid sea urchins.
Nice.
And, who knows how long it's taken them to do that and then perhaps to move on. So I thought that was very much worthwhile to have observed, and who knows? Maybe we'll get to see one on this dive en flagrante feeding, living up to a relationship that we've seen for a very long time.
Oh, yeah. Polymastia, yeah.
All right.
Right next to another one of those small euplectellids.
There's another stalked crinoid, it looks like, at 3 o'clock of laser. Oh, yeah. Keep going. I was saying there's a stalked crinoid. It looks like another one center screen. And there's one at 4 o'clock. Yeah, if I'm slowing you down from
and there's
making it to the top,
sideroid
please
right there.
tell me no and just keep going.
So we have both predator and potential predator, potential prey right there in the same shot.
Did someone call in? Guess not.
No, it's probably somebody hanging up.
Almost like right on cue to have those two animals adjacent to each other. Oh, that's a different sponge. The little white one right in front of us. At least I think it might be.
It's nice, soft looking.
Yeah, it's very soft. I saw some barnacles up there. There's a little,
Oh, there's a ACK hydroid.
Oh yeah, just to the right of the sponge is a yellow, large, single-stalked hydroid. Maybe a little solitary guy. We don't get a chance to look at them too often. Oh, it's very beautiful.
And a yellow cup coral on the left side.
Yep, cup coral.
The left side of the sponge.
See, this is yes, a beautiful cup coral.
And a crinoid. Different type of crinoid. That one's a cumatulate crinoid, unstalked.
Alan, I'm not used to seeing those large solitary hydroids in that yellow color. That was kind of cool.
That was kind of cool. And I was trying to get a feel. Sometimes the reproductive bits are right there in the up near the hypostome, the mouth area.
Mm-hmm.
Yeah. The gonophores. I think I saw them there.
Mm-hmm.
I'll have to rewind the tape later.
Exactly.
This is just Chris again.
Hey, Chris.
I was really gratified to see that we saw that sideroid feeding on that crinoid just as predicted. So if we see that again, could we get a longer zoom on that? Because that hasn't been seen very often, and I'm sure there's some folks who would love to have better data on it.
Yeah, sure. Can we look at that white at, now it's at 8, 7-ish? Maybe some living coral here. It looks like stylaster. Okay, it's stylaster or something. It's got awful long polyps now. I'm wrong. Coralliid?
This is a coralliid.
Coralliid.
Yeah. Corallium, probably.
Yeah.
Or, yeah, something like that.
Yeah.
I think I see a little cladorizid at the foreground.
Oh.
About 6 o'clock.
Was it pin cushiony?
No, it's the little sunburst one.
Oh, can we zoom back in? Where was it relative to the corallid?
It was growing out of coralliid in between a couple of branches on the side closest to you, so near the base, on the right side of the stalk.
Just a little to the right, he said.
Just above, I think that looks like a farreid or something.
Yeah, I see the farreid.
I see the claws of a critter of a.
Out of the branches.
This yellow, so that's a crab or a squat lobster.
More of those great hydroids.
Right.
No, just a little bit to the right, supposedly.
Yeah, I think it's going to be on the right side of that colony, what I thought I saw.
We may be too much on top of it now. That's all right. We can just push on. Boy, those yellow-brown hydroids are really...
Yeah, they're big, huh? A couple centimeters tall.
I get to have some fun figuring those out later tonight.
Mm-hmm.
So it looks like on the multi-beam we have quite a ways to go to get to the top.
I know we're trying to go.
If it is,
go ahead.
I know, but I'm just saying. The top, if it is living, is going to have a whole suite of new organisms to see. So behoove everybody to get up there.
Yeah, we'll go as fast as we can. I told him to step on it.
Right. I do think it would be good to take a water sample when we pause again.
On the slope here?
On the slope as well. So we'll get one when we get to the top too. But next time we sort of slow down.
Maybe Hexagorals at 3. Seeing the top of this mound over here a little bit, top of the ledge.
Yeah, at the beginning of the day, the mappers told us the shallowest part of the feature is 758 meters. So looks like we're getting pretty close.
Awesome. Yeah, I see a fish. Maybe a cutthroat eel or somebody's tail. Living in the dead standing coral.
Great.
Yes.
Thank you.
That was 3W, correct?
Yes.
Okay. We got a white gorgonian ahead. And the top of this feature just looks like continuous dead standing Lophelia and Lophelia rubble. Yeah, keep going.
So, Stefan, you've seen a lot of these coral mounds. Do they grow like towards the current? Is that kind of the way they do that?
So most of the living Lophelia is going to be facing into the current, which in this area is prevailing from the south because we're in the Florida current or part of the Gulf Stream. There's a recently dead one, I think. Yeah, the white. That's a gorgonian, but to the left of it is a fresh dead Lophelia, it looks like to me. Yeah, on a few occasions they find them on the north side, but that's because the prevailing current is actually a counter current that occurs at the deep depth. Most of the Lophelia reefs, the main coral habitat, living coral habitat, is facing into the southern current that's coming from the Keys area where we were a few days ago. That's because these corals do not photosynthesize the way that shallow water corals do. So shallow water corals have a symbiotic dinoflagellate called zooxanthellae that lives within their tissue, and that's why they're so bright and beautiful in the shallow water. And that is actually what bleaching is. So when the coral gets stressed out or is exposed to too much high temperature for an extended period of time, they'll actually expel those zooxanthellae out of their tissue into the water column, and those become free-swimming plankton again. And then when the coral de-stresses out or comes back to normal and becomes more healthy, those zooxanthellae will actually jump back into their tissue, and they will unbleach. Deep sea corals, oh, look at him carrying around his hat. How cute is that?
Oh, and he's just holding on to it.
Oh my
it's not
gosh,
growing
look at
to
his
him.
modified back legs made perfectly for carrying around such an adorable piece of clothing. So he's a little decorated crab. He's picked off some. He's not very good at holding the sponge, though, is he? You know, he has.
Yeah, exactly.
It's like
He's doing all right.
It's like an umbrella. He's like Mary Poppins.
Whee!
He's so cute.
I wonder if the sponge gets better flow that way,
I would
too.
imagine.
Mutual help with each other.
Yeah. That's so cute. He's like floating down.
It's like, get to the bottom of the hill.
Anyway, what I was saying is the deep water coral is so deep underwater that it's past the depth at which light can penetrate. So there is no photosynthesis at this depth, and therefore the corals would not benefit from having a symbiotic dinoflagellate that photosynthesizes within their tissue. So they are stark white. They do not have that symbiotic relationship with zooxanthellae, and therefore they rely only on food passing by them in the water column, and they use their tentacles to collect that food and feed themselves with. So they only have that one form of food intake. The shallow water corals do the filter feeding and the photosynthesis. So the only place they're going to get food is from the Gulf Stream in this area, or the Florida Current in this area, where the food is coming up from the south, passing over these mounds, and then feeding these habitats with particles and food that the coral can eat. So that's why the coral has a tendency to grow facing into the current, as opposed to on the back side of these reefs, which is the side we're looking at now. So we are headed south into the current, pointing forward.
We're pretty much at the top.
Yeah, and they're pretty much at the top here. So you can check out the other side of this mound, see if there's anybody living over there. But at some point, this was a humongous Lophelia reef.
Yeah.
Not a lot of standing dead up here. It's mostly coral rubble.
So, Alan, I'm looking at the multi-beam on screen 3, and it appears the reddish area is still, I can't tell the scale, but it's still to the south from where we are.
Okay. So there's more, it looks like, John, you think there's more height here to the south?
Look at screen 3, the multi-beam.
Let me get it pulled up.
And the reddish area, the shallowest, is still a ways, quite a ways. Look how far it is. It's a heck of a long way.
I see what you're saying.
Yeah, we got to get to waypoint 3, but no way, unfortunately. But do your best. Good luck.
Okay, that's great.
Okay, so Nav is saying we're about 134 meters from that next red high. So we are going to make that jump. There's a possibility we'll hit a little bit of blue water on the way. It's really thin here. It's like
this is like a little
saddle area.
Ridge, yeah. Since there's multiple highs on this. Oh, there's another
sea star, Stylosideris.
Sideroid.
Pencil
Does that one
urchin?
look different than the other one? Or?
I mean, have they long, thin? Maybe
Yeah.
they have the really long, thin pencil spines. Oh, he looks like he's eating a piece of bamboo, Carl.
He is. Since there are different tops of this mound, so it has sort of like knobs on it, I'm wondering if the active growing spot has changed over time.
That's very possible. I know some of the mounds off of the Little Bahama Bank, on the west side of Little Bahama Bank, just south of here, that Chuck Messing worked on, the lithoherms, that they showed Stephanie was saying, the coral's growing into the current, and the mounds tend to prograde into the current on the steepest side, and then the rubble is more of a gentle slope on the north side.
Yeah.
Hey, Alan, could we get a shot on that star before we leave?
I missed the star.
Yeah.
To the right of the sideroid you were just looking at.
Ah, thanks.
Yeah, it's at 3 o'clock now.
I'm not sure that was an actual star. I think it may have just been bamboo rubble. We can look at it again, though.
My apologies if it's not.
Yeah, I'm not sure what it is.
All right.
It looks funny from here.
People,
No, it's rubble.
it's rubble.
Tricky. It even got the starfish guy.
Exactly.
Well, when we were in Hawaii, we would see white. Oh, yeah, it is coral. Oh, see, dead starfish look like that sometimes.
Absolutely.
I mean, I was thinking that maybe the sideroid had been feeding on the starfish, but it's not. It is, by the way, a different sideroid than the other one.
Oh, good.
This one, I think, might actually be Stylosiderus. I think the other one might have been Siderus, just Siderus.
Okay.
But telling them apart is very difficult in the field. So, anyway, sorry. Thanks.
No worries.
All good.
Thanks. Oh, we know this guy. Chaunex.
Yeah, or Chaunacops.
He's cute. A little squeaker toy.
Squeak. That's not the rosy colored one that we saw last, that we've seen before.
Look at his little feet in the front. Our little pink feet. His modified pectoral fins. He does not look happy with us at all.
No. They always look grumpy, but they also are very cute.
They are cute.
He really stands out with his coloration.
Really bright, yeah, compared to everything else on this mound here.
Should we start a grumpy cat or a grumpy fish meme?
Oh, we totally should start a Grumpy Fish meme. I miss Grumpy Cat.
They come and go.
Aw. That was the cutest kitty ever. Here we got as high as increasing.
Yeah.
We still have a ways to go, though. Should just keep getting taller. So the slope's pretty steep here. Looks like you well above 45, 55 degrees.
So we're about 60 meters
So
from our...
So the big question is, why are some of these mountains totally dead, or, as far as coral, hard coral? And we do know in some areas there has been extensive bottom trawling by foreign countries and in the deep sea. And in the shallower sea, we've seen the Oculina reefs totally devastated by shrimp trawlers. And unfortunately, those same shrimp trawlers have access to the new deepwater coral HAPC off of Florida. They have a shrimp access zone that they've been given, and it's amazing how much destruction they can cause, dragging their weighted nets across the reef. It's just ridiculous that it's legal.
And we are actually outside the HAPC, so this is open to fishing.
So I continue to see that euplectellid, which Nolan Barrett has had a chance to look at more closely, and it's likely to be Heterotella, the genus Heterotella.
And I'm not seeing any dead standing coral. It's pretty.
No.
It's pretty all broken up here.
Lots of these little chrysogorgias.
Mm-hmm.
It's funny, off to the left there, it looks a little different. I wonder if it's possible, that you could have a big development of a Lophelia and the building of the Lophelia bioherm, and then, conditions are maybe change some, and then it's not conducive to Lophelias, but still the mound is,
still
creates
present.
all this. Well, the mound is present, but then also creates all this habitat for the diverse organisms we're seeing today.
I'm seeing a lot of rippling, which makes me think the temperature's mixing a lot here. And Lophelia only tolerates a max temperature of 12 degrees, and we're already at 10. So it might be a temperature thing, where it's being exposed to, areas of temperature that are too high for it to tolerate. But they would still be standing if it was a recent. If it was a recent die-off, they would be standing.
Right.
So it's had to have been a long time. And I don't see any evidence of trawl because I'm not seeing knocked over gorgonians. I'm not seeing trawl marks. That's not to say that's not the problem, but I don't have evidence either way. It is or isn't. I don't know who's out here, if people are out here fishing or not, on this ridge.
Right. But we don't see any.
But we are outside the HAPC, so if they were doing it, it would be legal. But so you can see the rippling of the water near the base, right near the bottom.
Yeah.
It's very ripply.
And Sirius up above has a different temperature on their CTD.
What are they at? 10.8? And 10.1? Is that what I'm seeing?
Yeah, they're like about
Almost
7/10
Yeah.
warmer right now. It was greater closer to the bottom too. There was a bigger
This difference?
differential. Yeah.
So we're about 759 meters.
Can you tell what the bottom current is?
I can find out. Do you guys know what the bottom current looks like? It's light. Like 1/10 of a knot, maybe? It's gone from west to east. And it's about 1/10 of a knot, John. Yeah, in this local.
Hey, this is Heather Coleman from Deep Sea Coral Research Technology Program calling in.
I think I heard someone call in. I was very faint.
Hey, this is Heather Coleman from Deep Sea Coral Research Technology Program.
Hey, Heather.
Hey, Heather.
You're very
Hi,
quiet.
what happened?
Heather, can you speak
Yeah,
up?
I'm using
Go ahead.
a landline, so I thought that would be better, but I can try something else.
Okay. Yeah.
Okay.
Is this any better?
Say something again?
Is this still pretty faint?
That's a little better.
Yeah.
Okay. I can try a cell phone if that would be better. I just figured a landline would work, but...
No, you're fine. I can hear you.
Okay. Well, I heard the word hapsy, and that got me all excited, wondering if I could call in and talk about hapsies a little bit.
Of course.
Excellent. So I know you guys are just east of the big hapsy in the area, that's in Miami, and thought it was exciting that the South Atlantic Fishery Management Council is using all of this information and past Okeanos cruise information from the last few years to inform their decisions in the near future about whether they want to expand the boundaries and by how much. So really direct connection from this work that everybody here is doing to management outcomes.
That's great to hear.
We've been providing presentations for the council for the last 2, maybe 3 years, for one of their advisory panels, and we always get so many questions about the mounds, about deep-sea corals in the area, about fish using the deep-sea coral habitat, anything you can think of, and so we always include some videos from these dives, and to start it off, to end it in the middle, videos and all the presentations.
Nice. That's good to know. I mean, the outreach here is amazing, right? The whole point of having this video footage streamed to the wider world is to A, educate people, B, entertain people, and C, to figure out where the heck we need to be with regards to protection and managing resources. And the South Atlantic Fisheries Management Council is in charge of that. So they're the ones who determine, based on input from not only scientists and footage like this, but also from the local fishermen, about where these HAPCs, or habitat areas of particular concern, should be located. And a lot of the work that was made, the current one, is actually from John Reed, my boss, who you've been hearing talk off and on today. A lot of that data came from his research over the past 40 years, working at a Harbor Branch Oceanographic with the Johnson Sea Link, and discovering these places that no one had ever seen before, and submitting that data and that information to the South Atlantic Fisheries Management Council, and helping them make the best choice for not only these reefs, but also the people who use them, and the people who want to fish near them or fish by them, while still protecting this precious resource, because our coral reefs only grow, these corals only grow at about 1/3 of a centimeter to a centimeter a year, which is the length of your pinky nail to the tip of your pinky nail, which is extremely hard to have these corals grow back if they are being damaged by possible trawling or other human impact activities. So it's really, really important that we know where these reefs are. We communicate that to the wider world and to the management councils and the people who live in these areas or around these areas, who fish in the water above them, or use their boats locally or recreationally, or use the local resources for what am I looking? The word I'm looking for. When people come down here and travel.
Yes, like tourism?
Yeah, tourism. Thank you. Look, I can
Yes.
remember 250 genus and species in a weekend, but I cannot remember what tourism is. Yeah, so, yes, there's huge impact on that. And the habitat areas of particular concern are areas that prevent the most of the time, it depends on the individual ones, but most of the time prevent impact on the bottom. So if you, maybe you can speak more to that, like how the HAPCs are formed and why they're formed, and the process that we go through to get them set up.
Absolutely. I can try. I wasn't working at NOAA yet when stuff in Miami was becoming a habitat area of particular concern, but it was in 2010 that it was actually implemented as a protected area. And I think that the process had started back in 2004, so it was a really lengthy, if I have that right, it was a really lengthy deliberation about what would be protected. And it makes sense. It's an enormous area. You don't do that lightly. So it's really careful consideration about where the boundaries would be. And the data were really limited, as they are now, but way more limited at the time. The boundaries were expanded in 2015, and so all kinds of things were taken into consideration for those boundary decisions. Like was just said, a lot of fisheries input, economic concerns, but of course, what's the habitat like? What needs to be protected to thrive and to keep becoming productive fish habitat? So since 2015. The habitat area of particular concern has been the size it is now, but there are exclusion areas for crab traps and for shrimp fishing. They're thinking about expanding along the eastern side of the boundaries of this habitat area of particular concern. So what I understand, somewhere near the area where you guys are now, maybe closer to the HAPC itself. I don't know. I don't think anybody knows yet how far they're thinking about expanding the area. But these dives will absolutely help inform those decisions. So the how HAPCs are created, it's very different in every council. So in the Deep Sea Coral Program, we work with councils in each region of the country. And the South Atlantic was really a leader in protecting habitat. They started back in the early 80s creating habitat areas of particular concern. Before that was actually part of the legislation. So before that was something within essential fish habitat, part of the Magnuson-Stevens Act. They pioneered HAPCs, essentially, and now the rest of the country is using it too as part of that legislation. So the South Atlantic really should be commended for all of the habitat protection work they're doing and are continuing to do.
There's the first live Lophilia right there.
John, I think it's Madrepora. Oh, maybe not. No, you're right. Sorry. Nice. Good job.
So our first big piece of living Lophilia, which is the species which is most of the framework, most of this rubble consists of.
Yeah, it's small. So that's good. That's good.
That's really interesting what Heather was saying about.
Yeah. Sorry, I didn't mean to cut you off.
Yeah. No, I will pause for Lophilia any day.
Yeah, definitely. It's a really small piece. It's about 10 centimeters, 15 centimeters wide, and the branches are very thin, which is why I thought it might have been Madrepora at first glance. But it's definitely Lophilia. You can see the widening tips.
I think it's really neat that the South Atlantic Fishery Management Council is the pioneer in protecting these habitats. That's really fantastic.
Absolutely. Yeah,
it is. And along with the Western Pacific Fishery Management Council, they protected their entire EEZ from trawling in about the same time period in the early 80s. But the South Atlantic protected for more than just trawling. They protected a lot of they protected the habitat from a lot of different types of seafloor contact gear, and that's even more valuable. You know, in the Western Pacific, there's some long-line fishing of concern. So in the South Atlantic, they're really taking a comprehensive approach to fishing gear management and seafloor protection, and it's benefiting us. Definitely, we get to see all of these great dives, protected areas.
Yeah. Awesome. Thank you.
Yeah. So I think we are at the top.
I'm not sure. A little bit
longer.
Bit
left to go?
Yeah. We really want to make sure we try and get a view of the south side of this if we can, because that's the highest likelihood of having a hot spot of living coral. Looks like maybe a little piece is standing dead in front of us.
It's interesting. I guess I was expecting we would see more live, especially by looking at how much rubble there was down below.
Definitely. It looks like there's some fresher dead here with white. See the whites? The white pieces? But they're not standing. They're all knocked over. Maybe they've been tumbled or rotated somehow. Yeah, we're at 750 meters.
It's very surprising. There's very little octacorals or sponges up here, which seems very odd for the peak of any pinnacle.
Yeah, definitely. And also no standing dead. Usually there's, if it was fresh dead, it would still be standing. And here it's all just rubbled out. Yeah, you can see if it's standing dead, it still has its structure. This looks like there's also some white fresh dead. If you guys can zoom in on the white, like a white piece of rubble for me. And look at the calyces. That's a good indication of how old dead it is. So here the calyces really aren't intact. There's a few. They just look like little holes, right? And some of them are a little intact. The browner ones. There's one polyp alive right there on that guy. At noon. Yeah.
Yeah.
But the brown is old dead, definitely. But the white looks like it's what we call fresh dead or new dead.
Yeah, there goes that hydroid again.
And you can see the calyces on the white ones a little better. They still have the ridges inside where the coral polyp lives.
Yeah, definitely. Those would indicate that that's pretty fresh dead as opposed to the brown.
Yeah.
It's pretty amazing that, we're just seeing the surface, and if you took grabs of the bottom, how many organisms live within the rubble, both standing and rubble. In the Okeanos, I found like 350 species of mollusks living within the coral. And, decapods, punculids, polychaetes, stuff you can see on the surface.
Yeah, you have to really pick the head up and, pick through it. If it's not slowing us down, we can do snap zooms on whatever you'd like, as long as we just, try and get to the end of this mound. I'm not sure what that is. A little dark manganese, ferromanganese coated thing with a little shrimp on it. Some small white gorgonians. They're pretty short, but all roughly about the same size.
Right. Like they all were same colonization event. There's
a corallomorph at noon. And a chrysogorgid. A couple of them, small chrysogorgid, little puffy gorgonians. That's the corallomorph in center screen. What's our off-bottom time? 4?
We've got about 20 minutes.
Okay. Here's a little patch of exposed sediment. Nizumia, rat tail. Look, there's a little tiny piece of sargassum at noon. It's floated down.
Floated down from above.
Mm-hmm. That's looking over the edge there. 45-degree slope, roughly. Kind of flattens out at the top, rounds out.
Rounds out, and still not seeing standing, live or dead colonies.
So far, we've only seen one head of Lophelia. You didn't see any at the beginning, right? Little yellowy
Looks
tan
like
sponge. Pacastrellid or something. We got a couple of solitary cup corals in there. But most of these gorgonians, the gorgonians are the most common thing I'm seeing that's living. And yeah, a couple of fish. They're sparsely branched, white, and all about the same height.
What's that?
That is a crinoid.
Oh, yeah. Crinoid.
That's 6 o'clock. There's pencil urchin. Some type of Stylosideris.
Stylosideris.
Sideroid.
So, Alan, does this mound, are we at the eastern edge of where we saw the mounds on the multi-beam?
Yeah.
Mm-hmm.
I think there's 2 or 3 more to the east and northeast of us.
Yeah, there are more.
But for the most part, this is, the eastern side of it.
This region. Yeah. Okay.
Is that guy feeding?
Sure looks like it.
On those little white colonies.
Mm-hmm. It looks like his mouth is up.
Oh, is that his mouth up there?
I believe that's his mouth.
So not feeding.
Aristotle's lantern is what they're called.
So it must be upside down.
Yeah.
Wonder if something grabbed it.
Yeah, and flipped him over. It looks like, though, there's some tissue off of this one, so maybe he just fell off. See the tissue on the upper at, like, 11? It looks like that's fresh ripped off there, so maybe he was eating right there, and then he kind of lost control.
That looks like a little small, the Africalistes growing there.
Yeah, Africalistes are really common species up here. Go ahead, John. There's one species. Oh, yeah, we do have like a bubblegum coral there, huh? 6 o'clock. All right. Yeah, right below. There you go.
Hey, this is Chris Ma.
Hey, Chris.
Hey, could I adjust one of your observations?
Sure.
That was, in fact, the anus you were looking at.
Oh, okay.
The mouth is on the underside. So wrong end. I mean, they're kind of top bottom. So, but if we were to swing around, not that we lantern and everything, you can see the teeth through it. The anus and the other bits are on top. So what you were looking at there was, in fact, what some people call bioerosion or feeding or whatever you'd like to do.
Okay.
But the next time you see one, maybe you could sort of get the lens on the underside and see, because I've seen these before, and you have the teeth eroding the tissue. So anyway, thank you. Back to
I
you.
thought I saw teeth. I thought I saw teeth.
There's another one. Let's take a look at it.
Okay, on.
Now that one looks like the top side.
Yeah, and it's on the other side is where it's feeding on the...
on the gorgonium, right?
Yeah. But I do think that looks similar, although the anus threw us, didn't it?
Sometimes the anus has spines around the edge also.
Oh, okay. Maybe that's what threw me off.
Yeah.
Let me go find a picture. Can't send it to you over the chat, but let me see if I can locate a picture of the spines that sometimes flank around that top side there.
Can you swing 9 o'clock for me? There's a white coral here. It might be living Lophelia.
Maybe there too.
Yep. That looks like living Lophelia to me. Sure. Yep.
And if all else fails, given how little time is in the dive, we could always just collect one.
Yeah, we could.
Of the
just throwing it out there.
Of the Sideris? Of the Stylosideris?
Anyway, thanks. I'll talk to you guys later.
Yeah. Water sample.
Alan, do any of the coral people need Lophelia for their genetic studies? I don't know.
Yeah, the Aspire campaign is collecting Lophelia for that, for their genetic.
They would want a piece out here.
Yeah. Yeah, go for it.
Perfect, thank you.
What do you want, dive 12? Yeah.
Thank you.
Hey, this is Heather Coleman from DHC Coral Research Technology Program calling in. Can you hear me any better this time?
I'm sorry, Heather, what'd you say?
I was curious if you could hear me any better on this one.
We can hear you a little bit better, but it's actually pretty much the same.
Fine. Okay. Well, I just wanted to ask John Reed a question, if I could.
Yeah, go ahead.
I was wondering what his most impactful experience with the council, the Fishery Management Council, is, like research that he's done and presented to the council. What made the biggest difference in their conversations or discussions about protecting habitat?
So the question to John was, did you hear her, John?
Not really.
Not really. The question, if I got it right,
go ahead.
Was what did you present to the council that you feel like made the biggest impact? Did I get that right, Heather?
Yeah, absolutely.
Yeah.
Okay. Hi, Heather.
Stylosteric?
So back in the early 2001, we had our first cruise exploring these deep water reefs with the Johnson Sea Link sub. And I told the captain, we headed offshore Florida off of Fort Pierce, and I said, go to 2,750 feet and go straight until we get up to North Carolina. And we went over 300 mounds of 15 meters to 100 meters tall. And on the way back, we started diving on them and documented that every mound was coral habitat, Ophelia. And then we did a lot of work off of on Miami Terrace and Portales Terrace. And so in 2004, I went to the South Atlantic Council meeting, and presented a 70-page report along with our video, and worked with the council to start drawing the borders for the HAPC. So at that time, we hadn't found any coral, Ophelia, shallower than 400 meters. So we put the west border along 400 meters, and then drew the eastern border along, just looking at the habitat, or looking at bathymetric charts and where it appeared to be high relief. And this is way before all the multi-beam was going on. So it took from 2004 till 2010 until we were able to make that the Deepwater Coral HAPC, which expanded in 2015. And now I believe it's about 17,000 square miles. And it's an incredible tribute to the council, and primarily to, again, we are being impacted by bottom trawling for shrimp, royal red shrimp in deep water, as well as long lines of crab traps for Chasion, and able to help protect these deep water reefs. And with all this new work with the Okeanos is incredible. You know, now we're seeing what's east of that eastern border that we drew 10 years ago.
That's incredible. Thank you so much for that work and for presenting it to the council and helping to make such an impact. Is this one of the first dives east of the border?
What was that, Heather?
Is this one of the first dives east of that HAPC border?
We did
second,
one in,
I believe.
Yeah, we did one in 2019-07 cruise, where we found 2 or 3 really huge mounds, and they were living Ophelia reef. And I don't know. I think there was one more on this that I'm not aware of, that I wasn't on.
You want to think about collecting now that we're up at the top and looking around and not really?
Do we have time?
Is there anywhere else for us to go? I think it'd be great to get one of these little.
What, John?
Yeah, I say go ahead and start collecting, because obviously there's no large coral heads up on top. So I just want to mention Andrea Quatrini and myself and several other people have a book or chapter in a new Springer series, Springer publication, on deepwater coral reefs, and we wrote a chapter documenting and talking about all the deepwater reefs off the southeastern United States, from North Carolina to Florida, and hopefully that book will be coming out shortly.
That's awesome. I don't see any live Ophelia to collect for the Aspire campaign, and I would hate to take the 1 or 2 pieces. There's a piece. Maybe we can get a tiny little piece of that. I'd hate to take too much just because it's the only one that's out here.
I know.
I don't want to decimate it to nothing. Do you want to get a piece of that?
I think it'd be a good idea.
Yeah.
It'd be a good idea to get a piece of the genetics.
Yeah, can we go and get a tiny little piece of that coral that's right in front of us? So that's a big no, huh? Too late? You want to just quick snag?
What's the top one, the top choice there? The
Ophelia for genetics, yeah.
Mm-hmm.
Yeah, just a tiny, tiny, tiny piece. I don't want you to decimate all that's left up here. It's less, is it so tiny? Yeah, maybe we shouldn't then. A piece on the side. There's a beautiful 5-armed...
oh, yeah, let's take a look at that.
Star next to it. Not star, what am I? Feather star. Crinoid.
Yes? No. Ophiuroid? Oh, no, is that a crinoid?
Yeah. It's red and pink. Yeah, I know. How about the one on the side? Oh, it's very fragile. The one on the side, if you look down a little bit, the one over here at 3 o'clock has some living tissue on it. We just snag that piece. If you can say no too. I mean, I'm okay. If you're not comfortable. So 05B. Yeah.
So I know they've looked at the genetics comparing Lophelia from the Gulf of Mexico, the southern part of the Lophelia reefs off of southern Florida, and over on the eastern Atlantic, to look at the genetic connectivity. So it would be good to get one for this easternmost part of the Lophelia reefs in the western Atlantic.
Yeah, agreed. I know Cheryl Morrison did a big paper on that, looking at the genetics and its genetic connectivity between the Gulf and the Atlantic Lophelia species.
Yeah, that's definitely fine. We'll deal with it on ship easily.
Going to do our last water sample snap before we take off?
Absolutely. Sort of a replicate for the top here.
Yeah, go for it. Niskin?
So where do you plan to dive tomorrow and Sunday?
Tomorrow is a midwater.
Yep.
And then Sunday we're doing a Million Mounds dive.
Million Mounds, yep.
Yeah, so hopefully we'll get a good reef on Sunday.
Will that be like west of here?
Will that be? Do you know where the Million Mounds line is?
Yeah, that is going to be west of here.
What kind of depth is that one?
We have a pre-dive call after this, right? I'll find out. John, hang out.
Okay.
Yeah, I'll find out for you. Just give us a few seconds.
Is that within the HAPC?
I believe it's going to be within the HAPC, yeah. So it probably comparable depth and probably be able to compare what's living, what's inside and what's outside.
Right. Okay. And no dive on Monday.
No, Sunday's our last day. We pull into port Monday.
Yeah. Okay.
Okay. Did we already get the water? We're calling that 5W?
Yes.
Did you call that
Yes,
5W?
I called it 5W.
Okay, thank you.
Yeah,
Did you have a time for Emily?
20:52.
20:52 and 50 seconds. Mm-hmm.
Yep. And that final million mounds dive is going to be to 813 meters. So that will be on Sunday. And tomorrow we'll be doing midwater transects. A little bit further out easterly on the Lake Plateau.
Mm-hmm.
Southeasterly from here. And we'll be getting down to depths of about 1,050 meters.
So deeper than this one? Are you talking about
tomorrow,
the midwater?
The midwater,
okay.
yeah. Now, the million mounds is going to be just about the same as this.
But inside the habitat area
Yeah.
of particular concern. So be interesting to see if there's a difference in, or if we're in a similar situation where we've got a coral rubble mound versus a living Lophelia reef.
It's almost
There you go. It's in. Oh, I see. It might be caught in the hose. All right, it got picked up.
Yep.
I think it's a choker.
Yeah, right. You might have to spit it out into your bucket. Into the bio box. There you go.
Is it hacking up a bone?
It's in. Thank you.
Yay, success. That
All
was
right.
good work.
Now it's time to leave the bottom.
Sadly, I'm not ready to go, but I get it.
There's a lot to do.
Yep.
Okay, nice dive, guys.
Thanks for
Thank you.
tuning in. And helping out.
Yep. Thanks very much, John.
Six B. Yeah. Thank you. All right, so that's the end of our dive today. Thank you so much for tuning in, everybody, and everybody landside science. Thanks so much for your input and your commentary. It really helps us out a lot. Also makes it a lot more interesting.
Yep.
And we talk so much, it's very nice to have breaks sometimes. Just to give you a recap, we are 152 miles from shore. Off the coast of Florida, east coast of Florida, and 34 miles outside of the habitat area of particular concern that is dedicated to protecting the Lophelia reefs in this area. So we are outside that area. The depth at bottom that you reached was about 820 meters at the base of this mound. And the base of the mound was Lophelia rubble, looks similar to this, with the, normal regular species that are in there.
Yeah, we saw a lot of plexaurids and other small gorgonians and a couple primnoids, and then actually quite a lot of primnoids, right? And then but just a couple bamboos, on the way up. And we saw this euplectellid, either heterotella or dictyalis, something like that. Quite
glass
frequently,
sponge.
Glass sponge all the way up.
With its little commensal married couple of shrimp.
Yes, we did spend a little time on on a commensal shrimp. And we saw some nice sideroids. So we saw a few different kinds of urchins. We saw a couple different sideroids.
Ariosoma,
Yeah,
the
and then
pancake
the
urchin.
Ariosoma, exactly.
Only a couple of small colonies of living Lophelia coral up here. Most of it is dead rubble. We're not really sure what the reason for that is. It could be a couple different things, but
we encountered some corallomorphs, and I don't recall that we saw any black corals, but we did see some of those little pom-pom spiky cladorhizids at least once or twice.
And they're a type of carnivorous sponge. Max min depth was about 750, 745, 750 at the top of our highest local high. We were facing south most of the time, but even when we got to the upper edge, there was no living Lophelia except for a couple tiny little pieces. We did our 5 water samples for environmental DNA, and we collected 1 Lophelia for the Aspire campaign, which is going to be used to look at genetic connectivity within the species across the Atlantic. So we are planning a midwater dive tomorrow. We're going to have our post-dive call shortly, so if you want to be involved in that call, feel free to stay online or call in right now, and we will discuss that off the air. And then the following day is going to be our last dive of this cruise, and that's going to be on the Million Mounds area within the habitat area of particular concern at roughly the same depth we were doing today. Hopefully we're expecting to find a giant Lophelia reef. So if you were not as excited about this one.
We did find a giant Lophelia reef.
Living Lophelia reef. If you were not as excited about this one, that one should be the bee's knees. So anything else?
That's good.
It's good enough for now.
Yeah, I think that covers it pretty well.
Stop listening to me and Ellen blabber for a couple hours and tune in tomorrow. Thanks so much for tuning in. It means a lot to us, and we really appreciate your interest, and everybody's input is very helpful, especially when I cannot remember my critters' names today.
I think that's okay.
It's been a rough day today.
It's all good. It's all
right,
good.
so if you're wanting to chime in on the midwater dive tomorrow, hang on the line. We'll be right back.